Now we're going to talk about what happened when you don't have oxygen. What happened during cellular respiration when you don't have oxygen? So for example, when you work out running so fast that there is not enough oxygen, what is happening in these cases? And maybe you have heard about this word fermentation. And if you have eat cheese or you had a beer or a wine or anything like that, you know that fermentation is very important. And when you graduate from biology, this is a place maybe you will be working in some type of industry that uses fermentation. So let's dig into this topic. So we're going to talk about fermentation and anaerobic respiration and how they enable to produce ATP even without using of oxygen. Most of the cellular respiration require oxygen to produce ATP. And why? Because we have here all these molecules that are collecting these electrons, that it will transport the electrons, and these electrons will later will combine with oxygen to form water, while the hydrogen protons will form ATP through this redox reaction. However, if you don't have oxygen, you cannot have this redox reaction. So the only ATP that you will form, it will be this one. The majority of the ATP will not be formed. Now I want that you look again at this, and we just said that if we don't have oxygen, what is going to happen with the electron transport change? So I just explained that, and I told you that there was a redox reaction, and without oxygen we cannot have this redox reaction, so this is not going to happen. Yeah, so what is going to happen? That in the glycolysis, as we saw before, the pyruvate is going to take a different path. It's not going to go to the mitochondria, it's going to stay in the cytoplasm, in the cytosol, and depending on the organism, if in yeast is going to produce ethanol, or if in muscles is going to produce lactate. So these two cases through fermentation. So what will happen with the electron transport change? Just to recap, that it will cease to operate. It's not going to work anymore because there will be no more redox reaction. To start, again, your textbook gave you this wonderful video that summarizes all the process of fermentation. I want that you watch it, and I'm going to again highlight some important points that can help you to understand this process in more detail.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are two types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
Again, highlight some important points that can help you to understand this process in more detail. While you have oxygen, the glycolysis couples with the process in the three process in the mitochondria, pyruvate oxidation, the Krebs cycle, and the electron transport change. However, if there is no one of these process, in the absence of oxygen, what is going to happen is the glycolysis is going to couple with the fermentation in order to produce ATP. So remember, if we don't have oxygen again, we're going to take a different path, and this path is the glycolysis is going to couple with the fermentation and is going to produce ATP. Let's talk about the regeneration of the NAD+ and how this is done. I just remind you that during glycolysis you go from glucose to pyruvate and you produce ATP, But however, the most important part is that you start getting NADH molecules that will be used in the electron transport chain. When you don't have oxygen, you need to regenerate this NADH. You don't need it anymore. And how you do it? You do it by doing fermentation. So both in mussels or in the yeast, the fermentation is going to regenerate this NAD plus, so you can keep doing glycolysis and this process can continue. So to remember from this slide, this process is very important, that because during the glycolysis, the fermentation regenerate the NAD plus, and this is an important input for the glycolysis, so the glycolysis can continue. To remind you that maybe a question of the test could be the common types of fermentation, and I know that we already talked about that and you watch it in the video. And here I just remind you that these two types, they have to regenerate the NAD plus, and they are the lactate. This is the product, so it's the acid lactate fermentation, and the other one is the other product is the ethanol. So the type of fermentation will be the alcohol fermentation. And if you see this in your textbook, the reactions in more detail, here you can see in both cases the regeneration of the NAD plus, and here you have the pyruvate, and you can form the alcohol here, in this case ethanol, as we have it also in the diagram. And here you go from the glycolysis, and from the pyruvate you produce lactate, and this is the lactic acid fermentation. So to remember, there are two common types of fermentation: the alcohol fermentation that produces ethanol, as we have here, and the lactic acid fermentation that produces lactate. Alcohol fermentation is very important and is involved in many processes. So for example, one that you remember that alcohol fermentation by yeast is used in brewing, wine making, and baking. Now I want that we study in more details how fermentation takes place. And you go from a glucose that have 6 carbons to an ethanol that have only 2 carbons. And here you can see that you have 1, 2, and 3 carbons. So you go from glucose to pyruvate. And here, to remove the other carbon, you release a CO2. So you here, the second step is to release a CO2 molecule, and later what you do here at the end, as we talked before, is to regenerate the NADH, and in this way you get ethanol. So to remember, the pyruvate is converting into ethanol in 2 steps. So here I told you that there is a release of CO2, and the other step that I told you is the regeneration of NAD plus, and then to be used in the glycolysis. Now let's talk about the lactic acid fermentation in more detail. And here again you go from a glucose that has 6 carbons, and now you see here lactate has 1, 2, and 3 carbons. So different from the alcohol fermentation that we studied with the ethanol, here we end up with a 3 carbon molecule, and this is going to have consequences. The first consequence is that you are not going to release CO2, so there is no release of CO2. However, as we said before, you still regenerate the NAD plus, so when you produce the NADH, later you're going to regenerate it to continue doing glycolysis. So let's recap the thing that we said. I told you that the pyruvate is reduced by the NADH, and here you can see what is happening. And I also told you that the forms lactate as an end product, as you can see here, and as a result, as you have 3 carbons, there is no need to release CO2. Now let's talk about lactic acid fermentation. And here you see two products, yogurt and cheese, and those products are a result of lactic acid fermentation. Also, humans in their muscles, they can produce lactic acid to generate ATP when there is absence of oxygen. And in the next slide, we will study how from glucose you can go to produce a lactate molecule. Now, I'm just going to compare the fermentation and the aerobic process. And in both cases, they have something common. Both they use glycolysis and they oxidize glucose to harvest chemical energy from food. So this both in fermentation and aerobic respiration, this take place. In addition, the second thing that I want to remember is that the NAD plus is the oxidizing agent that accepts electrons. And in both cases this happens. Remember, in the aerobic respiration, this is going to go to a transport electron change, while this one is going to be regenerated to continue doing glycolysis. Now we're going to compare the final electron acceptors and the ATP production. So while in fermentation you get the final acceptor or the lactic acid or the ethanol, in the aerobic respiration will be oxygen. So this will be very different. And in the ATP production they are also very different. While the fermentation produces very little ATP, the aerobic respiration produces 16 times more ATP.